Welcome to MedTech Speed to Data, a KeyTech podcast. I'm your host, Andy Rogers, VP of Business Development at KeyTech. Each month, me and a KeyTecher are going to interview a MedTech leader and talk to them about the critical data-driven decisions they make in their programs.
Hello, and welcome to a special episode of MedTech Speed to Data. I'm Kelly Parker. I'm part of the business development team at KeyTech, responsible for cell and gene therapy and our life science tools market, guest hosting for Andy Rogers here today. We'll be focusing on some exciting automation topics within the precision medicine spectrum. Thinking about like high throughput automation in drug discovery, as well as lower throughput automation in the cell therapy manufacturing realms. I just want to welcome all of our guests here, Blair, Nova, and Daniela, and I'm going to pass the mic over to them so that they can kind of give us some background on their personal background, where they are now, and kind of what they're focusing on these days.
Great. Maybe I can take it here. Daniela Hristova-Neeley. I'm a partner at Health Advances. I spend all my time in life science tools and services. So on the tool side, anything from basic research all the way to pharmaceutical manufacturing. And on the services side, I spend most of my time in CRO, CDMO type pharma services, things that are really closer to the science and the technology, in part because I'm a scientist by training and I really like the science behind everything. So great to have this discussion today.
Blair Morad, Senior Director of Engineering here at Cellino. I've been involved in cell therapy for, I think, close to 20 years. Early days of cell sorting for cell therapy applications and really instrumentation development and consumables for use in single-use patient care.
Awesome. I can go next. Nova, Syed. Almost 15 years in the healthcare industry, from doing bench science in molecular biology to management consulting, followed by healthcare tech, now coming full circle in the biotech life sciences space. Most recently, I was part of the founding team at an SF-based startup called Mekonos, which used silicon nano-needles to make gene-modified cell therapies more accessible and more affordable. We raised $30 million in venture capital and landed our first large pharma partner. Prior to Mekonos, I was an in-house advisor at the Ontario provincial government, where I shepherded reimbursement policy decisions on health technologies, ranging from surgical robots all the way through to remote cardiac monitors. Excited to be here today.
Thanks, guys. We're definitely looking forward to a good discussion today. So I guess we'll kind of get into our first topic. So Danielle is going to kick us off. And I guess the first kind of thing that she'll be discussing is what's the role of automation in high throughput screening, and what is the driving need for automation in high throughput screening?
Thanks, Kelly. Yes, a very exciting topic. From our consulting world, we spent a lot of time working with scientists trying to figure out how to do go-to-market strategies and anything related to commercialization of new products. And automation and high-throughput screening are two topics that are very much top of mind and have really revolutionized how scientists do drug discovery and development by really enhancing efficiency, accuracy, throughput, you name it. Several factors have really driven the interest in automation and high throughput from a market perspective. First is really just the increasing nature of biological systems that have been studied and the number of drug targets that are being researched as it relates to drug development. Second is really the demand for faster drug development at lower cost. I'm sure we all hear it, but everybody is looking for things to be faster, better, and cheaper. And that has really driven the need for automation and high throughput. And last but not least is really the need for high quality reproducible data to meet regulatory standards. As we know, regulatory standards continue to increase. And as we speak in cell therapy in particular, obviously there are a lot of requirements as it relates to data traceability, accuracy, precision. So these are really the main themes that are driving automation and throughput. And a couple of examples as it relates to cell therapy where automation and high throughput screening have really made big differences: cell line development and also assay development. In cell line development, obviously we are looking for high performing clones that have reduce contamination, that increase we can increase really the throughput as we do the cell line development for those technologies, and automated systems have been really fundamental technologies as it relates to cell line development there. And similarly for assay development, whether that's assays for potency testing, where we are looking activity of different cell therapies, so whether that's for purity or stability testing, again, having an automated high throughput system really helps with faster assay development, keeps with better data quality, more accurate assays, which obviously helps with the development timelines for those assays and also with the requirements from a regulatory perspective. So all in all, I would say automation and high throughput screening have made a really big difference as we do drug discovery and development, and continue to be a very much top of mind from a user's perspective. Obviously, everybody would like to have a fully automated system that has a really high throughput. And Kelly, I'm curious to hear your thoughts as you help companies as developing the actual technologies and the systems, what are the challenges they're wrestling with, and how you help those clients.
Yeah, so definitely an interesting question for instrument design perspective. I think we're constantly working with our clients to work through how to balance the level of throughput with the instrument complexity, the cost, as well as the time to market. We're really having those conversations of balancing the need to have the perfect instrument with all of the features versus having the minimum viable product instrument with features that are good enough and kind of gets the product to market faster. I think also de-risking your core tech early is makes a big difference when you're developing this high throughput system. You need to make sure that it's working on the small scale. Is the technology even feasible? And then does it continue to work at the larger intended scale? Sometimes it does work at the small scale, and then when you try to scale it up, it isn't translational. So it's definitely important to get that information as early as possible. We like to break things early here at KeyTech, so we can pivot and move appropriately.
If you were to give one advice to a technology developer of an automated system for cell therapy, what is this advice you would give?
I would say it's important to start your instrument design considerations as early as possible. You need to understand the market need and kind of ensure that your high-risk subsystems are de-risked early on so that when you're integrating, integration can move as smoothly as possible, and you're making sure that that automation is going to work at scale as expected. Just because the higher throughput and the more complexity you add, it just adds more risk.
Great.
So I guess kind of hearing from industry folks, we've got a couple questions for you guys. How do tool developers approach high throughput instrument development? And I'll leave that open to Blair or Nova, whoever wants to tackle that one first.
I can go first. I mean, you touched on the critical piece on the core technology. It was really, really important for us as we thought about what our tech stack would look like for high throughput applications or low throughput. If either modality of cell therapy, it's really critical to de-risk upfront and make sure, like you said, from an integration perspective, it can go as smoothly as possible. Never perfect, right? Because we know there will be risk embedded in just putting multiple modules together. But certainly if there's a consumables component, there's a workflow component, surface chemistry, material compatibility, all of those factors come into play at an even larger scale when you think about integrating into your customers' workflows as well as tools developers. For us, that was a complaint that we heard repeatedly from large pharma partners, is that they would pour millions of dollars into very niche instruments that didn't truly integrate into their workflows, and ultimately ended up, being shelved in a corner. And as tools developers, that's the worst case scenario. We want our systems to be used. Blair, I don't know if that resonates with your experience in the industry.
Yeah, for sure. I mean, we've certainly, especially when you're partially automated, when you do some of the automation and then you integrate it with other people that they're doing other pieces of the automation. I just remember a situation where, we had tubes that were being used for, aseptic connections. And so, you just tube weld it, put a new tube on. Well, the tubes we're using were not used by all industry standards. Some people using different tubes. And so their tube welders are different. And so it's just a lot of thinking through those details when you're integrating with a bigger ecosystem of things. It would be lovely to own all of it but it's also a lot of work to do that. So trying to, making sure you're plugged in with as many people in the market as you can be, and understanding where things are going and how things are working is something that also I think we spend a lot of time thinking about.
Yeah, definitely getting those interfaces right is tough, and you want to make sure you're approaching that on the front end and not figuring it out on the back end. And same Nova with the usability. That's huge for adoption ultimately. I guess what instrument design questions and trade-offs do tools companies contemplate when it comes to the high throughput instrument design?
Blair, you want to take that one first?
Sure. The tools trade-offs, I guess, design considerations. I mean, I think a lot of it is, are there existing systems out there that we could leverage, and where and when does that make sense? For a company that specializes in optics, for example, we probably want to, own that and design something for it. But if we need an incubator, I don't think we're going to start developing an incubator, right? So finding what our core competency is, and then making sure that we're sticking to our guns on that, and then leveraging what's out there is definitely a design consideration when we go into it. I think it's pretty tempting to kind of think about it all and trying to come up with solutions. But especially when you have, really sharp people that know how to solve really hard problems, making sure that we're solving the right ones.
Right.
So that's something that, is constantly discussed and, refined in the early stages. Yeah.
Definitely, I'd agree with that. I think our process for setting requirements really started with a very broad scope, and reining in all of the kind of intellectual curiosity that a multidisciplinary product team can have is just part of the process. But certainly you have to be ruthless when it comes to prioritizing what is your differentiator. In our case, it really came down to the cell types. Which cell types are the hardest to transfect using existing conventional methods, electroporation and viral vectors, and where are the challenges when it comes to cargo size, cargo type as well, and using the intersection of those two dimensions as our anchor point, if you will, because otherwise, there's lots of cool cell types and lots of cool cargo types that could be awesome to deliver just from a scientific standpoint, but not necessarily commercially. It may not necessarily have the commercial traction that we need to have to build a viable business.
Can you guys kind of share a little bit of your learnings as you've gone through these programs? What worked or what didn't work well on previous high throughput automation programs?
I think I can speak to the trade-offs a little bit more. My main learning, having seen companies reach out for reimbursement or market access from the health system side of things, automation was a huge factor for building system capacity when it comes to diagnostics, for example.
Mm-hmm.
But as a tools developer, the trade-offs between time, cost, and scope, quality was always table stakes. It could never, especially when it comes to if you're, depending on the applications and the use cases, if it's leading to a therapeutic, it has to meet those quality metrics and scale for the therapeutics developers as well. So oftentimes we would, towards the end of our development cycle for our MVP, we ended up reducing the scope in order to be able to hit the time and cost metrics, because those are also limited. But quality was always table stakes for us.
I think similarly, the testing side of things and the verification, validation work that goes into it, even like a mini form of those,
Mm-hmm.
the sooner you can do them, the better. I find that we're often in the situation where we have these really grand ideas about how we can get this thing on the market and so on and so forth. But then when you get to actually testing it, it becomes really hard, right? And so, thinking about those requirements, are they testable? Those are some basics, right? But then just like thinking about like, oh, well, how do we even know that we did this right? Like how big of an end do we need to have to actually know that we did this correctly? And going through that, you start realizing, wow, this is going to take months, or could take more than that, to really get to a point where you feel confident, and that, the regulatory side of it is confident that this is something that's there. Right. So, Yeah, I think that's something that life lessons, I guess, in their previous existences, previous companies I've been at, where, we sort of rushed, especially as a startup mentality, sort of like, we're going to rush and do this. And then you realize that when you actually get there, it's like, okay, there's a lot to do. It's just something that to put that in the forefront and get people thinking about, like, how are we going to prove that this is going to work? Not just, this sounds really great and it does all the things we want it to, we've done it once and it works great. How do I know that it's going to be right? And, what kind of surrogates can we use besides cells and, like, patient, samples, to prove this out? And how fast can we do those tests? Like, can we accelerate those tests so we get better results, or quicker results, and be able to iterate quickly and come up with other solutions to it? That's something that, I've certainly tried to bring more into my organization and how we operate.
Maybe if I can just jump in with one question. I'm curious, since you, Blair, and Noah, you both spoke about quality and the importance of quality and how you need to consider, what do you do first and what data you need to generate to prove that the technology that you're developing is success. I'm curious, when you make, like, how you think about trade-offs and how do you think about what the actual end user is asking you in terms of features and capabilities of the technology that you're developing versus as you think about the internal development and what you should own. I mean, Blair, you spoke to some of the things that you would like to own as part of the technology versus things that you partner or integrate with. I'm just curious, like, from a developer perspective, how do you make trade-offs? Because obviously in an ideal scenario you have it all, but there is no such ideal scenario.
Yeah, totally. I think as an early stage startup with a lot of emerging professionals on our team, the default kind of mentality can often be to go in house, right? To develop, to build from within, because there's an immense gratification in going from 0 to 1. But our kind of philosophy, which took a lot of training to build, was really coming down to, again, what is our core technology? Where are we differentiated? And has someone else, built, whether it's assays, workflows, automation modules before outside in the industry? Who are the experts? And if we're not the experts, then there's no shame in asking for help outside of our core company. That's, I think, philosophy or a mindset that has to be trained over time and through experience. So certainly, yeah, there were trade-offs, and some of those lessons are learned through experience, right? After having gone through 1 or 2 cycles. We had that experience for our consumables development, for example. Our initial kind of approach was to go in-house, but given how long things were taking and the evidence bar that we had to maintain, because we had for every project, we would have a customer at the end of it. And that kind of standard couldn't be compromised. So some of it is learning from experience. Some of it is going direct to the experts that we know, folks like Keytech, folks like Waynamics out in the Bay Area, both from an instrument and a consumables development point of view. Our development cycle just went from 1 to 5x after we had gone with experts. So once I think the internal team sees the value, then you've have conviction across the company. What about you, Blair? What was your experience like?
Yeah, it's similar. I think there's always this, like, struggle between iterate as fast as possible internally and get to a solution versus, really define what you want and then do a transfer to somebody that is an expert that can understand this and can move forward. I've always been a fan of, like, if you have that sort of, like, partition wall, it's not a very high wall. I mean, you still can reach over and talk to the person, but it's high enough that you're sort of, you take a moment to think about what it is that you really want and then articulate that. I think there's a lot of benefit in just taking that time versus, like, oh, I'm just going to go in the shop and make something and I'll come out and we'll try it and we'll do this and and just kind of keep iterating. It kind of creates this opportunity for us to just take a moment and think about it. And especially when you're working with the right experts, they know how to ask the right questions and kind of curate the information in a way that's like helpful to like innovate and move forward on it. Yeah.
That's a great point, actually. If you haven't defined the problem clearly enough, then no matter what kind of expert you have, won't necessarily be able to help you as much as they could if you had spent that kind of internal time really honing in on where you want to focus and knowing what kind of help to ask for, right?
Definitely. We love to hear that people will stop and take the time to like plan through that, even though it's hard sometimes when you're so excited and you're like raring to go, but to kind of think through like exactly what does success look like at the end of this and how are we going to get there? Like what are all the things in addition to like the design that need to get done, like the testing and speaking to the right parties and things like that. So kind of moving on to the next topic, more specific in cell therapy. Daniela, you kind of talked about the role of automation in high throughput screening and research, but what is the role of automation in cell therapy manufacturing specifically, and kind of what tools are required for cell therapy manufacturing?
Yeah, interesting, because we spoke earlier in research and development, we always talk about high throughput, right? And automation is always driven by let's do more and get more as much data as we can. When we go to manufacturing, I think it certainly depends on what type of cell therapy or gene therapy we are manufacturing. As Nova mentioned earlier, it's really about the cell line, the cell type. But automation has somewhat different purpose. Again, part of the reasons, if we speak in particular about the autologous cell therapies, right, we would like to have flexible systems that are scalable as we scale up and go to later stages of development and potentially commercial. We would like to have better quality control, right? We would like to make sure that there is less interference with the manufacturing system and less human interaction so that we have a better quality control. And we also have better data traceability so that we have things to demonstrate to regulators as well. And then again, cost is everything. Even in manufacturing, we know that many of those cell therapies are pretty costly for the healthcare system, and so there is constant push and desire to automate and integrate and make the systems more cost effective. So automation certainly has the potential to reduce the cost. And again, we, in autologous therapy in particular, we talk a lot about closed systems. How do we have everything integrated in a single system so that you have the cell therapy comes or the after leukapheresis, the cell, if you will, starting material comes into the manufacturing system, and then you at the end have a very well QC'd drug product, which is again the cell therapy product that is going to the patient. And these closed systems certainly have the potential to decentralize how manufacturing is done today and make things closer to the patient and potentially give better access to patients in particular geographic areas. So automation has a lot of purpose here, and it's a main focus as from a user perspective, from the manufacturing engineering perspective. It's a big priority, and I'm curious, Kelly, again, how you guys help in KeyTech the developers design better, more automated cell therapy manufacturing systems.
Yeah, so I think some considerations that we like to have in mind when we're starting in these engagements in this space is we need to understand the science, very similar to when we're working on an IVD program. You need to understand the assay. Here, you need to understand the biology and have that pretty established so you can start designing your automated manufacturing steps early to ensure that they're going to be compatible with the cells. And then there you can kind of start scaling it. And kind of to reiterate what Daniela said, it needs to be flexible. You need to be able to easily change those parameters very early on based on testing, but then be able to lock things down, so to say, for manufacturing scale up, which will help you transfer from bench to process development suite to ultimately the GMP suite. And that flexibility is key. It allows you to move quickly and efficiently by responding to your data, but still progressing your design. And designing a flexible system is definitely a challenge in and of itself, but is important in this market, I think. And then you need to consider usability from the beginning. These are super complex processes, generally with pretty substantial user interface. And I think bringing automation in, you kind of want to take a lot of that burden and kind of reduce the use error potential with the automation to make it more reliable and repeatable, especially when you have that one patient sample that you're putting in. It's very, very valuable to get your therapy product out. So definitely curious to hear kind of like from Nova and Blair, how you guys are considering information when you're approaching instrument development in the cell therapy manufacturing specifically.
Sure. Yeah, it's there's a lot of places to get information on how to what is the right way to do things, or the challenge we've always had, and this not just where I am now, but historically, is knowing the what the partner or what the application is, and how is that being used. On the plus side of like an R&D type space, you're sort of developing your process, so the bioprocess associated with that. And so the benefit of that is that you have a lot of flexibility there, right? Because you can change automation on the fly. You don't have a whole lot of GMP to go through to do that.
Mm-hmm.
But the challenge is that that instrument itself is quite complex, because it has to potentially do all sorts of things that we just didn't think about, right?
Mm-hmm.
But when you move to GMP, or when you move to like a manufacturing space, like you're sort of locking yourself into a process, right? So that's there. That is the process, and so the automation equipment can be simplified. But knowing which pieces to simplify, sometimes you just have to kind of get to that point with the partner or whatever the application is that you're working with. But it's a lot of work to get things working right. I'd say one of the things that I've noticed is that with this the automation equipment, the challenge that's within GMP is a lot of, even though there's automation, there's still a lot of users involved in it.
Mm-hmm.
Just generally, like, got to bring the media up to temperature. Someone's going to bring it in there and like warm it up, and then they're going to add it to the system, and then they're going to do this, and then so there's all these people sort of swarming around it.
Mm-hmm.
And so you have this automation thing inside, but you still have a lot of people involved in doing it too. And so the human factor is still really important on it. I think the sort of the magic wand of like, I dump cells on one side and I get finished product on the other side. It's just not quite there yet. So, so it is something that we're moving there slowly but surely. But it is, there's a lot of low hanging fruit on the automation side to improve on. But proving that works reliably every time is sometimes the hard part.
Yeah. I find it definitely challenging in this market because cells are unpredictable. They're living things. They don't always operate or react the way that you expect them to, which is a new challenge for us, I think.
Yeah. Years ago, I was doing cell sorting, and we were, as a surrogate for cells, we were using, fluorescent beads to help us understand how things were working. And the scientists would just yell at us and say, beads are not cells, and I've got that ingrained in my head. Whenever I'm doing an experiment from an engineering perspective, like these are not cells. Like what we're doing right now are not really cells, and they're going to behave differently. And we should just, but we just don't want to burn through all this technology or this development without, with the cells and the patients involved as well, because that's costly, and it's unpredictable. So
it really is precious material, right? Especially if you're dealing with primary cells. So having a systematic way of, going up one level from beads to a cell line to then primary cells is really beneficial when it comes to development. I think the digitization is another kind of unlock that automation helps achieve when it comes to cell therapy manufacturing in general. That's one of the reasons why cost of manufacturing is so high, is so much human intervention still expected, if not required, still expected, just given how much, precious material there is, and it's going back into a patient. So we're not quite there yet where it's fully automated, but I think as we head towards decentralized manufacturing, that trend is still, the momentum is still there. Multiple companies working towards that. So it's very exciting to see. I think one learning for us was around the quality metrics, right? So what are the actual critical quality attributes that our large pharma partners or medium-sized biotechs, whoever the customer is, what do they care about as they're taking a drug from R&D to process development through clinical trials, etc. What's the target profile, product profile? Even though we ourselves as tools developers are not developing the drug product, understanding what those metrics are, how they're defined, and how they change from one cell type to another is hugely beneficial as you're thinking of system design, workflow design, etc.
Yeah, absolutely. That's a big challenge in this field. I feel hearing and talking to other people in the industry that tool developers are facing with those various cell types, and they aren't reacting the same, and you need to understand all the differences. So thank you for bringing that up. I guess, so as you guys are working in your cell therapy companies or previous cell therapy companies, what were some of the instrument design considerations and trade-offs that you had to develop there that might be different from considerations for a high throughput system?
Size was a big one for us. Initially, it was a lot of negotiation in terms of what the footprint could be, how much tolerance there was, especially if we're thinking long term about a deployed instrument in a GMP environment. That's also costly space that our customers have to pay for, right? But at the end of the day, I think where we landed there is if the value created from a certain tool, whether it's ablation technology like Blair's or transfection technology like ours, if the value created out of those tools is significant enough, the space will be made for it because the pain points are that strong when it comes to quality, when it comes to delivery. That's one top of mind example that comes to mind for me.
I think a major trade-off that's always on the top of mind is time to market, and maybe not necessarily market to the patient, but like the market to our investors, the market to the next level that we're trying to get to is always this backdrop, right, to everything we're doing, and so trying not to necessarily feed into it too much, but at the same time realizing that it's important, right? And so I think one of the things that we spend a lot of time on is both where I am now and what I've done in the past is like what is the most important thing that we have to prove that this as we're developing it that once we get to that threshold, then we're ready for the next thing. And you talked about earlier about MVPs, and I think that's something that's really important in this space. And I have examples of like throughput's really important, but also the performance or the quality of the data we're getting out of it. And so there's this constant tug of war of like, do we make a system that is high throughput and is able to sort of process a lot, but maybe not so great, or do you want to go slower? And so those initial customers, which one do they care more about? Do they care that we actually are getting good quality data and high purity, high yield for like sorts, for example? And we'll get the speed later, right? And trying to get that into the market, and then realizing that sometimes especially as people are building their protocols, like you sort of get locked into a certain device within their protocol, then like trying to add that in, it becomes another layer that they have to prove out that's working. And so there's just a lot to think about. But I think, balancing the need to show that things are working the way we want them to is something that we sort of prioritize as far as our technology that we're developing.
Definitely. We stand behind that. De-risk early and understand your user to make sure that you're building the right product for your intended market, and getting out there and talking to them and testing with them throughout the entire process makes the whole iterative process much easier. I say easier. We all know that there's fun when you get feedback.
Less hard.
Less hard. Yeah, that's it.
Yeah. Better to get it early than late, I think. So I guess if you guys had to start all over again at your cell therapy companies, what potentially may change the decision-making processes that you've had to make? Or I guess, are there different things that you would do differently approaching this fresh again?
That's a great question. I think being in the early stage startup space, there's so many things that we would have. I'm sure I can speak for Blair. We would have done differently. For me personally, I think the stepwise approach in terms of like what we prioritize, the wet workflow became really, really important for us. So starting with the chemistry, the protocols in terms of what types of reagents and the material compatibility, starting there, in addition to our core technology development, which was always the first priority. Developing scalable fabrication processes for silicon nano-needles is no easy feat. So I think like while we were focused on that, the wet workflow side of things tended to get deprioritized. So that would be not like number one, that we would have more resources placed on the chemistry side, in addition to consulting with tools developers, technology developers that have lessons learned to share with us, right? That's the other thing. We're in this big ecosystem of life science tools, instrumentation, and in addition to our end customers, the pharma, large pharma players. But there's a lot of lessons learned that can be shared if there's a true sense of community. So those would be like big lessons learned, takeaways, in addition to many other things. But I'll let Blair go next.
The one thing I was thinking about when we talked about having this podcast was like, we're sort of a disruptive technology within a disruptive science. Mm-hmm. There's a lot of things happening, and a lot of pieces are moving, and people are learning new ways to do things. Applications are popping up all over the place, but maybe they're not far enough ahead. You're working with some partners that have really good science behind it, but they actually don't have a commercial path. You have people that are, may have a commercial path, but not a very viable product. So it's sort of like a lot of these things that we're trying to navigate as well. And I would say, early on, we sort of as a startup, you just want to kind of cover all your bases on your idea, and think about all the things. And I don't know whether if we went through and sort of focused on one area for more time, whether we would have learned more about, like, say, our functionality, like the core functions of our process, like what is actually really happening at that cellular level. We didn't really, we're doing that, but we keep learning about it and all the things that are happening, because cells are unique and they're different. And so we're constantly learning about it and like reevaluating, like, oh, maybe this is the way, what's happening here. And I think it's all part of the journey. Like, I don't think I would necessarily change any of it. I think it's the right things to do. But it's just getting everyone on the same page of like, we just have to be patient about it. There's a lot that we're going to ebb and flow on. And, it's a roller coaster of a ride when you're developing something. And sometimes it works really well. And other times it's like, not the best day. Right. So, you're just kind of riding with it.
Most definitely. And yeah, this market is kind of like the Wild West. It's come a long way, but it still has a long way to go. But it's very exciting to be in it, and seeing these changes happening. Danielle, I want to pass it back to you and see if you had any additional thoughts or comments or questions for the crew here.
I mean, really, it's great discussion, everyone. I appreciate that there are a lot of challenges, and as Blair and Nova put it, it's we doing science within a very innovating, novel technology like cell therapy. So there are a lot of considerations, and automation is certainly important, but it's not the only thing that is going to solve how we do cell therapy manufacturing or cell therapy development. And obviously the customer, it's always an important consideration in my mind, especially because I spend so much time in consulting, helping companies commercialize products. So from my perspective, it's always important to hear what the customers have to say. And I think Nova put it well together, that they work with their partners to figure out what the end user actually really needs as they think about developing some, if there is anything else Blair and Nova can share.
Yeah, I think on that front, the commercial traction front, a reflection that I've been having lately is that the product market fit doesn't just happen to you, right? You have to put a product out there, learn from the market, and there's risk involved in doing that. A lot of trade-off calculations go into making those types of decisions, and how you're going to go to market is part of that. But unless you get feedback directly from the users, directly from the customers, who, by the way, could be different stakeholders altogether, if it's a large enough company, unless you get that feedback, you don't have really the data to be able to iterate to the next cycle, right? We all come from a scientific background to a certain extent, but I think when you're in business, applying that scientific mindset is harder to do because there are real world ramifications when it comes to investment and company viability, all of that stuff. But it's really important to iterate by getting real feedback from the market. I've found it to be really useful.
Yeah, I think there's something said about the fact that there's a lot of startups doing cell therapy. In the sense that it is a disruptive field. There's a lot going on. It's hard to gauge what the market is really saying. You're sort of threading a needle. A lot of my experience has been, you find that you want to find that sweet spot of a partner where they're not quite ready for manufacturing because you're not anywhere near that. But you also don't want the person that's like so early on they have no idea what they want, right? Mm-hmm. So you want to find the so where do you fit? Like when is the right partnership going to happen? And the solution, at least from what I've seen, is just talk to as many people as possible, and maybe something will come out of all that. So cast a big enough net, you'll catch the right fish. But I think that's something that is always the challenge in this market. And the engineering stuff is fun and the science is great, but at the end of the day, that commercial path and where does it fit is always the hard part, it seems, with finding that right spot.
Awesome. Well, I really enjoyed the conversation today and enjoyed catching up with you guys and kind of hearing a little bit of your war stories from personal experience. And I hope to catch up with all of you at a conference or when we're in the area this fall. But I want to give a special thanks to Daniela for helping narrow down the topics for discussion today. And thanks, Blair and Nova, for joining us on this podcast today. It's been a pleasure.
Great. Thanks for having me.
And thanks for hosting us, Kelly.
Yep.
Awesome. Thanks, guys.
Take care.
Bye.
Bye.
Thanks for tuning in to MedTech Speed to Data, a KeyTech podcast. Join us each month for more ways to get the right data faster to inform critical decisions. Find additional resources on our website, keytechinc.com. If you like this episode, don't forget to subscribe, and please leave a review on iTunes whenever you listen. Thanks.